गुड मॉर्निंग स्टूडेंट. नवयुवक शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली welcome you in e-learning platform prepared by myself Dr. Shubhangi Prabhakarao Chavan, Assistant Professor and Head Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded semester pattern curriculum under choice based credit system CBCS for undergraduate program, Faculty of Science and Technology, subject Botany, year BSc first year, semester second, paper number third, Bryophytes, Pteridophytes, Gymnosperms and Paleobotany. Now student, in last lecture we studied general characters and anatomy of Marchantia. Today we will discuss sexual reproduction in Marchantia. Let us start Marchantia. Sexual reproduction in Marchantia is oogamous. All species are dioecious. Male reproductive bodies are known as antheridia and female as archegonia. Antheridia and archegonia are produced and special erect modified lateral branches of thallus called antheridiophores and archegoniophore respectively. Further, growth of the thallus is checked because growing point of the thallus is utilized in the formation of these branches. In some thalli of Marchantia palmatic and Marchantia polymorphia, abnormal receptacle bearing both antheridia and archegonia have also been reported. Such bisexual receptacles are called as anthrogonous receptacles. Now, next, the diagrammatic representation of Marchantia. Marchantia gametophores. In figure first, A, thallus being antheridiophores. Now see, we can see antheridiophores. Figure number B, female thallus bearing archegoniophores. We can see archegoniophores. And lastly, figure number C, transverse section of gametophores. We can see air pore, air chamber, rhizoids, gore, clube. That is the diagrammatic representation of Marchantia gametophores. Now, next, internal structure of antheridiophores or archegoniophores. Its transverse section show that can be differentiated into two sides, ventral sides and dorsal sides. Ventral sides has two longitudinal toes with scales and rhizoids. These groove run longitudinally through the entire length of the stalk. Dorsal side show an internal differentiation of air chambers. Now, next point, antheridiophore. It consists of 1 to 3 centromere long stalk and a lobed disc at the apex. The disc is usually 8 lobed, but in Marchantia graminata it is 4 lobed. The lobed disc is a result of created dichotomies. Now, next, LS through the disc of antheridiophore. The disc consists of air chamber alternating with the heridial cavities. Air chamber are more or less triangular and pour on surface by a N pore called ostiole. Antheridia arise in acropatial succession, that the older near the center and youngest at the margin. Now, next, the diagrammatical representation of Marchantia antheridia. Firstly, first figure, figure number A, ventricle or longitudinal section passing through disc of antheridiophore. First of all, we can see air pore, ostiole, air chamber, antheridial chamber, antheridium, air chamber, stalk, thallus, groove, that is the ventricle or longitudinal section passing through the disc of antheridiophore. Next, figure number B, mature antheridium. We can see antheridial mother cells, jacket, stalk. This is the diagrammatic representation of Marchantia antheridia. Now, next point, mature antheridium. The mature antheridium is globular in shape and can be differentiated into two parts, stalk and body. Stalk is short, multicellular and attached the body to the base of the antheridial chamber. A single layered sterile jacket encloses the mass of anthrocytes mother cells, which metamorphosis into anthrozoids. The anthrozoids is a minute rod-like bioflagellate structure. Now, next point, development of antheridium. The development of the antheridium starts by a single sacrificial cells which is situated on the dorsal surface of the disc, 2 to 3 cells behind the growing point. This cells is called antheridial initial. The antheridial initial increases in size and divides by the transverse division to form an outer upper cells and a lower basal cells. Now, basal cell remains embedded in the tissue of the thallus, undergoes a later further development and form the embedded portion of the antheridial stalk. Outer cell divides to form filament of the four cells. Upper two cells of the four called filament are known as primary antheridial cell, and lower two cells are known as primary stalk cell. Primary stalk cells form the stalk of the antheridium. Primary antheridial cells divide by two successive ventricle division at right angle to each other to form two tiers of four cells. Each a peritoneal division is laid down in both the tiers of four cells, and there is formation of 8 outer sterile jacket initial and 8 inner primary androgonal cells. Now, next the diagrammatical representation of Marchantia, development of antheridium. Now, figure A, Successive stages in the development of antheridium. Antheridial, we can see antheridial initial figure number B, basal cell and outer cell. Figure number C, primary stalk cells and primary antheridial cells. Now figure number D, we can see stalk cells, and figure number E, primary antheridial cells, jocket initials, jocket cells. Now figure number F, we can see stalk, jocket cells, antheridial cells. Now lastly figure number G, mature antheridium. Mature antheridium, we can see antheridial mother cells, antheridial cavity and osteols. This is the diagrammatical representation of Marchantia, development of antheridium. Now jocket initials divides by the several anticlineal divisions to form a single layer of sterile antheridial jocket. Primary androgonial cells divides by the several repeated transverse and vertical division, resulting in the formation of larger number of small androgonial cells. The last generation of the androgonial cells is known as androchoid mother cells. Each androchoid mother cells divides by a diagonal mitotic division to form two triangular cells called androcytes. Each androcyte cells metamorphosis into an anthrozoite. Now next, the diagrammatic representation of Marchantia spermatogenesis. Firstly, diagram A, antheridial mother cells. Figure number B, we can see formation of antheridiocytes. Figure number C, as it is same. Figure number D and G, stages is spermatogenesis. And lastly, figure number H, a single anthrozoites. The diagrammatic representation of Marchantia spermatogenesis. Now, student, thank you.